Good evening, class. One person should just respond to me, then we start our lesson. Okay, so sorry. My network was having a little issue, and I have to set up 3 devices. So, to those of us we are meeting today for the very first time, I'm Michael Adubuache, teacher at YAX. I'm sure most of my students know me as at now. So I'll be going through with you the second lesson for week 5, and that will be movement of substances in and out of a cell. So maybe. Okay, now you can unmute your microphone. Since most of you have confirmed, you can see me now. You can rather mute your microphone. Mute your microphone, please. We are far behind time. We are 14 minutes into our time. Mute your microphone. Mute your microphones. All right. Thank you very much. So we started by talking about movement of substances in and out of a cell. So we look at the structure of the cell membrane. That was yesterday, sorry, Sunday, when we talk about the cell structure, the cell membrane structure, the function, and membrane proteins, and a lot of things. So where we reached was the movement of substances in and out of a cell. So, one or two persons will just tell me anything you remember from a previous lesson. One or two. Anyone who is ready. So I'm sure now most of you can see my screen. So you can raise up your hand. Kwabena, the person using Kwabena, your microphone is on. Please mute yourself. Okay, I've done that for him, so don't worry. Anyone, if there's any question from previous lesson or anything you remember in the previous lesson, you can raise up your hand. I'll call you then. We'll start today's lesson. I'm sure all of you can see my screen as at now. A hand is up. Who is that? Stephanie also talk to us. Yeah, Steph Science 9. I think so. Stephanie, of course. Okay, thank you very much for correcting me.
The last one is we learned about the need for movement of substances in and out of the cell.
Oh, okay, and what did we say those things were? Let me change my screen so that it doesn't seem you are reading from my screen.
Okay. We said that one is for cells to obtain nutrients
Mm-hmm.
and create waste substances.
Yeah, yes. Very,
That's what I remember.
very good. Those were the things that we talk about at that time. Very, very good of you. So I think we can just start since we are far behind time. We can just start our lesson. So we we talk about the need for substances to enter the cell. Now we ask ourselves, how do substances do enter the cell? That is the next thing we have to talk about. So actually we have let me say 3 main ways in which 2 main ways, in which substances can enter in and out of a cell. And the main one is we have the active transport and passive transport. So now that we know the reason why substances have to move in or out of the cell, we have to know the means in which substances can enter in or out of the cell. So I explained that passive. So in I think economics, or let me say business line of action, we call something passive income. Then we have active income whatsoever. When we talk about passive transport, it means there is no active involvement or energy is not being used. So passive income, for instance, I buy a vehicle and I give it to a rider, or let me say a driver to drive it. So the driver will make sales every day. That one I'm getting a passive income. If I invest in a mutual fund or I buy some stocks every month, if I do get some return from the investment or the stock rises and I sell my stock, I get profit. That is passive income because I am not the one actively involved. I am not working with the money to get the income. So that becomes a passive income. So in the passive transport too, you are not requiring energy for you to move the substances in or out of a cell. With the active transport, there will be the need or use of energy to move the substances in and out of the cell. So that is the 2 main ways whereby we can use energy, and some other instances we don't use energy, and that will be referred to as passive transport. If there's no energy being used, and it becomes active transport if energy is used. So our number is not more than 100, so I will not utilize the live session here. When it's up to 100, someone should notify me. I will then share the link with you, then people can join and have a feel of the live session even as we are on the Google Meet platform. So movement of substance in and out of a cell, we have two main types, that's the active transport and passive transport. Under the passive transport, so these are the two main ways, but aside that, we can talk about a third one that we call a vesicular transport or bulk transport. So I will explain that shortly to you. So we have the passive transport, as you can read here, energy is not required. So the two main types of passive transport are diffusion and osmosis, which we will tackle diffusion today. And if we are likely to have enough time, we will talk about osmosis and active transport as well. So under the active transport, energy will be utilized to push that substances or that molecule in the cell. And basically, why are we using energy? Because we are going against a concentration gradient. So since we are going against a concentration gradient, energy will be required to move such substances in or out of the cell. But under osmosis or diffusion, the molecules themselves are moving. And when the solute molecule is moving, it is diffusion. But when the solvent molecule, particularly water, is the one doing the movement, we say osmosis. I will explain very well when we get to the time. We talk them one by one. So if you are here, you can also go to the WhatsApp page, the administrators. You can tell them that we have started, and I'm sure some left because we're having some technical hitches. So you can go there and inform them that we are live and we have started rolling the lesson already. Thank you very much. So, and the other one is called vesicular transport or bulk transport. So here is like, sorry to say, a cargo car. So here some substances cannot just move through normal molecule or particle movement because they are in a larger volumes and they require a bulk transport, or let me say a cargo car, for them to be moved in and out of the cell. So normally they can enter the cell through something that we call endocytosis or exocytosis. Endo means inside, so and cytos from the word cytoplasm. So here we've not talked about amoeba now. We'll talk about how amoeba engulf its food very shortly in the next month or so. That is lesson, and the ecology, we will talk about that. So assuming this is the thing that we want to transport, bulk transport. This one is not just a single molecule. We cannot enter through the cell by just simple active transport or maybe diffusion. So what happens is that the part of the cytoplasm will open up, then engulf this. So that is called endocytosis. So endocytosis, it will move closer to the cytoplasm, and the cell membrane will now, because we say it is fluid, because of the fluid mosaic model, it can open up, allow this unwanted substance to move out of the cell. That becomes exocytosis. But phagocytosis or endocytosis, you are now engulfing it and taking it in. Don't worry, there's an image I will show to you in this slides. I'm sure by now most of you have read about that, so you know much about that. That is the vesicular or bulk transport. So a large amount of molecules will be transported or substance across the membrane, and they form a vesicle. So a vesicle is whereby the cell membrane of that cell that we are talking about, this membrane will form a structure around the molecule. Then once it enters it, the cell wall, or sorry, the cell membrane of that cell has now engulfed the substances, and we call that in a vessel. So you don't see this mouse again. It will be a part of my skin covering this mouse for it to enter my hand or my palm. So that is called vesicular formation. So that's one function of the cell membrane when it comes to vesicular transport or bulk transport. I will explain that very shortly. So we have two main types of passive transport, and we have the diffusion and osmosis. Can someone help us to record the definition for diffusion? Someone should help us. I know this one, most of us know. Raise up your hand to make it interactive. That is why. Yeah, a hand is up. Let me check the person. Sorry, it hasn't appeared on my screen now, and I don't want to be switching devices. Someone can just raise up the hand. You tell me. Yeah, Salome. Yeah, yeah. Talk to us, Salome.
The diffusion is the net movement of molecules from a higher concentration to a lower concentration.
I want you to add one last thing to get 3 out of 3. You've really done well, but add the last phrase, then you get 3 out of 3.
Diffusion is the net movement of molecules of a region of high concentration to a lower concentration.
Let me add that last bit for you. The first one was perfect, but the second one, add it.
Through a semi-permeable membrane.
Hey, so sorry. Let me come in and help you. That even nullified the 2 points that you had. So
I have remembered.
tell us.
Diffusion is the net movement of molecules of a region of high concentration to a lower concentration until the molecules are evenly dispersed or distributed.
Very good. In your definition, it's from, but you said of. Is the movement from. So next time, remember you don't confuse of and on. So you've done very, very well. Let me just reiterate what she just said. She said it's the net movement of molecules from a region of higher concentration to a region of lower concentration until the molecules are evenly distributed. But because of how you even started, you might not even necessarily add that one because it said it's the net movement. So I do understand now why you were not adding it. If you say it's the net movement, it means that a there is a balance. Are you getting it? The moving out and moving in has now been balanced. So those who add the net movement, I'm sure the marking scheme will not penalize you for adding the last bit. But always, as I advise my students, it's better to write more, over explain than under explain. So some books will say it's the movement of substances. They will not bring the net movement from a region of higher concentration to a region of lower concentration until the molecules are evenly distributed or until the molecules are evenly spread or there's a net movement. So you can add these things to make the definition better. Thank you very much. So the key words here is the movement of molecules. So it's the solute molecules that are moving, and they are moving from a higher concentration to a region of lower concentration. Some want to be so specific. What is the higher concentration? Some want to add higher solute concentration. That is over explanation. We will not mark you down for doing that. So the solute is rather making the movement. But when you come to osmosis, something else happened. Now, someone else, if your microphone is up, if your hand is up, show it again. Sorry if I couldn't see your hand up. You can raise up. Yes, let me check those hands that I have so I can call them in order. Okay, Chloe Asibo, yeah, so your hand is up. Let me give you the question now. You are defining osmosis. Help us define the term osmosis. Chloe, if your hand is still up, talk to us. Chloe, if you can hear me, unmute your microphone, then you talk to us. Tell us the definition of osmosis. I'm sure she can't hear me, so anyone who is ready. Someone said my network is scratching. I don't know. If you can hear me very well, give me a thumbs up. Maybe that is that of Salome. Just to be sure it's my network. If you can hear me very well without any network hitches, give me a thumbs up. If you're also facing the same challenge as Salome, let me know so that I will be sure it's my network for tonight, not that of hers. Okay, maybe I said maybe I said it's okay at your side. If it's not okay, give me. Someone said she's right, sir. Okay, so two people are having the same issue. But check it now. If it's okay now, let me know. Stacy, so it's okay. So now some are saying so, some are saying otherwise. So please, those who are saying they have that issue, leave the meeting and join outright. Leave and just join within some few seconds, and let's check to be sure it's not my network or that issue. Someone said, now Salome, the one that complained, can hear me clearly. So you're having an issue, leave the meeting, then you join straight away so that no one takes your position. Sorry, there's an echo. Okay, do you all hear it? Yeah, it's my other device. And that is where you are seeing me from. So I don't know. Let me take it very, very far away. Because I want to do the recording myself, I have to mute some microphone and unmute some. That's why even the recording there was an echo over there. Okay, I'm praying the echo will not be that much. Okay, if everything is okay, let's start. So sorry, we are having these technical hitches. Okay, there's a hand up. Salome, talk to us. Osmosis. We are trying to define osmosis. Salome, unmute your microphone and talk to us. Harry Tosu, what if you are ready, talk to us.
Sir, please, osmosis is the movement of water molecules across a semi-permeable membrane from a region of higher concentration to a region of lower concentration.
Okay, you've done well. Any other person to help us? So we okay. So can you say it again so that I can tell you where you need a little bit of corrections? Can you repeat? Then I'll later call Janelle.
Osmosis is the movement of water molecules across a semi-permeable membrane from a region of higher water concentration to a region of lower water concentration.
Yeah, now I think it's perfect or near perfect. You made that very well. Janelle, let's hear from. Hey, it's okay. Not until. Just say across a semi-permeable membrane. Here we don't normally say the until, so don't worry, don't add that one. What you add here is across a semi-permeable membrane, and that is perfectly fine. So thank you very much. I think that was Harry. So Janelle, your hand is up. You can talk to us. When I call you, you have just 3 seconds to talk to us, so at least it will help us all. Janelle. Okay.
Osmosis is described as the movement or passage of water molecules across a membrane from a weak solution to a strong solution through a semi-permeable membrane.
You said from what solution?
From a weak solution.
Okay. To a strong solution. Very good. Very good. That is perfect. There is a name there. Efia disturb. Okay. Now you can disturb us, Efia.
Sir, please, I'm coming.
Okay, Efia.
Osmosis is
Your network, I think you started on a very good point, but the network was having some hitches, so I couldn't hear you very well. If you can come again, then we can continue.
Osmosis is the diffusion of a substance through a semi-permeable membrane from a
Oh, okay. From the way you are going, I'm sure you are very right. So thank you very much. You've all really done very well. These are definitions for osmosis. So as we said, osmosis is the movement of some will say solvent molecules, not solute, solvent, or some can even just say water molecules from a region of higher concentration. From a region of higher water concentration or weak solution to a concentrated solution. So the key point is water is moving from where it is more to where water is less. And if water is more, it means the concentration is very weak, or the solution is having a weak concentration. When water is less, then it means the concentration is very, very strong. So water is moving from where it is more to where it is less. So these are the key terms you have to look out when you are trying to define osmosis. I hope you are all okay. I am hearing some beeps. Let me try. I don't know why tonight my setups are giving me some feedbacks. Alright, so let me continue. We've talked about the osmosis already. Let's now look at the diffusion proper. So we have two main. So we see over here, we never talk about using energy in the form of ATP for any other thing. Now we have diffusion. So in diffusion, atoms or molecules of gases. So I said the other time that flow uh, solutes are rather doing the movement. So it's a substance rather that is moving, and it could be a gas or a liquid, but it shouldn't be the water itself. When the water itself moves, then we are talking about osmosis. So here we will look at diffusion in real life and some in that occurs in cells. So molecules do move randomly, and I'm sure you talk about that under chemistry and physics. That is how substances, molecule be moved. Gases, molecules, a solid, and the other molecules. You understand them better in chemistry, and I think physics will talk about that. So when these bombard or they collide with each other, that causes them to spread about because they gain momentum and kinetic energy as they collide and they evenly spread. So when heating comes in, we'll use that to explain why heating can increase the rate of diffusion in cells and other substances. So therefore, diffusion can be defined as the net movement of molecules from a region of higher concentration, or from a, sorry, it should be off here, off should be here. So sorry, don't have it here. From a region of higher concentration to a region of lower concentration until the molecules become evenly distributed. So why? There is a concentration gradient. There is a difference between the concentration. So once you have something like that, there will be a movement just to balance things out. So if we are having a higher concentration here, a lower here, the sun will move from this side to downstream so that it become a little bit equilibrium. Someone says you can't hear me. If someone can hear me, let me know. If you can hear me, give me a thumbs up. If you can't hear, give me a thumbs down. Let me know whether it's only here. So I've been telling you, if you have an issue, you can just leave, check your network first, or you can leave the meeting, then you join very shortly. If you can hear me, give me some thumbs up. Let me know she's not the only one with me. I'm waiting for your reply. You can unmute your microphone and let me know whether you can hear me.
Yes.
Okay, thank you very much. Okay, thank you very much, Harry. So I think one day, yeah, thank you all. So maybe another, yes, thank you very much. Now, thank you that you can hear me. So the one complaining, do the needful, leave and join very shortly. So let me continue. So maybe what I have to find one person as a Google Meet rep so that anytime anyone has a complaint, you will testify to let me know. I have to keep some device away from it so that the echoes and those things will not be much for our meeting and recording. So that is diffusion. There's a net movement of these molecules to where it is low to create an equilibrium so that they are evenly spread. That is why sometimes when they spray a perfume, if there is no other perfume, you smell the scent of the perfume very, very fast. But if a lot of you are having their perfumes on and you put up a new one, because the concentration gradient is not that wide, you don't smell it very fast or very well. You only get to smell it well once you are having that as the only perfume around. That means the concentration gradient is very low. So for instance, let me add, maybe you're having two muddy water from the same container. If you put them together, you don't see any difference. But when you add a clean water to that dirty or muddy water, you see the difference as they are meeting. So it's like there is a pipe that has ruptured, and that pipe is now flowing into the earth or into the soil or the dust. You see that over there, you see the difference because the water is very clean. There is no water around, so you see the sharp difference between the water coming from the pipeline and the soil. But once the soil now becomes engulfed with water and you see a muddy water and another fresh water is joining, you see the difference, the contrast become to lower to the point that you have a very lower concentration gradient because they are moving with a dirty water around the same time. So when you join two dirty or muddy water together, you don't feel the difference or sharp difference. But when you add a very fresh water to a muddy one, that is when you feel the sharp differences over there. So that's concentration gradient. When it's very higher, diffusion is very very fast because they are moving very fast to create the equilibrium. But when it's very low, like 2 to 3, it's not that wide, so you might not even sense it. That's why if you're having 3 or 4 people using the same spray and you just open yours up, we might not smell yours. No one might even notify this a new spray being sprayed over there, a perfume being sprayed over there. But when we are in a very let me say a foul center place or an airy place without any perfume and you open all yours up, everyone over there will get to smell the scent. So I think that a lot of things. And now let's look like some real life process of diffusion. So at this point, let me try to ask from you, give us there's something we have in living cells and also real life application. I think in our examination it was in living, okay, not you, I think I asked the question somewhere else. So it could happen in living cells. We have examples in living cells and some are not living cells. So just give me an example of diffusion. This one is a general one. If you can tell me one in just life application or what occurs in living cells, raise up your hand. Let me call you 1 or 2 people to make it interactive. Example of diffusion, whereby substance move. A hand is up, but I haven't seen it yet. Harriet or Wusu, your hand is up. Talk to us, please.
Sir, please, when lifting is dissolved in hot water.
Okay, so explain why it is diffusion, just to be sure of what you say.
Okay, so the tea particles in the bag, they evenly distribute the water. Diffusion.
Okay, thank you for making it diffusion. That is all. So that is the I don't know what they used to make that later. So the lifting in the tea bag, the substances or the molecules from it will now dissolve with water. So now that it has been dissolved in water, you will see those colored substances or solution formed moving inside the water. So if you are the step, let me call you right now.
When you add boiling water into noodles, sir, please add them.
Can you explain that? This Yeah, if you are, let's try
The
to.
water
Mhm.
mixes.
Okay, if you are, let me come in. When water is doing the movement, know for sure it's osmosis. When water is doing so, this cannot be accepted. But if you tell us that maybe you added, you see, we have.
Sir, no, I mean when you add,
I'm listening, talk to me.
sir, when you add boiling water into dry noodles.
So what is doing the movement? I'm trying to explain things to you, so just listen to me very well. What is doing the movement in this case? What is moving?
Osmosis.
Is the water that is moving? Is that not so? Help me to help you. So if water is doing the movement of here is osmosis, and I don't think this can even qualify very well to be osmosis. So the water, yeah, the water is now entering the noodle, so that becomes osmosis. So we can take that. It is very similar to water entering the root noodles, but not your instant or let me say Indomie noodles. So if water enters root nodules, that is called osmosis. So it looks similar to what we are talking about. That is better called osmosis because water is doing the movement. So thank you very much for bringing a very similar life application question, but answer. But that is for osmosis. So when we get there, we can use that as a very good example. Chloe, Asibu, your hand is up. Talk to us.
The fluid that dissolves in water.
Very good. Or someone can say potassium permanganate. When you are applying glue, what we call that glue, you see that it just dissolve and spread evenly. Though before at the start, it doesn't spread evenly around the inside the water molecule, but it takes some time. If you want it very fast, what do? You stir. You just stir it and everything dissolve very faster. And there's another thing that you can do. If you want your potassium permanganate or your glue to dissolve faster, let's tell ourselves what the things that we can do. Let me call Wajem. Your hand is up. Answer this question before you bring your answer. Wajem, you've lowered your hand. Okay, Chloe, if you are back, you can talk to us. Someone is using a blue. You don't want to dissolve it. What do you do so that it can dissolve faster? Tell me the things that you can do. Chloe,
Then you
I'll
can
call you.
stir.
You can stir very good. So stirring. Keep this thing in mind. You can stir. What else? There's something else that you can do.
Then break into smaller pieces.
Okay, break into smaller pieces. You can do something like that by breaking it into smaller pieces. So that is you are okay. Any other thing? So stirring, breaking to
Then
pieces,
adding hot water.
adding hot water, or someone can say heating instead of adding hard water. You realize like these will be the reasons or the condition that increases the rate of diffusion. So any other thing that you can talk of. Mm-hmm. Okay, people have raised their hand. Let me call them. Janelle, I called you, but you didn't say anything. If you are ready, talk. If not, I will lower your hand for you. Janelle.
The spread of insecticide spray or perfume in a room after spraying it in one corner.
Okay, so she has given another example of diffusion. That's very, very good. So when you apply that spray over there, the insecticide spray, you smell it across over there. But if, for instance, I have applied heaven and someone want to apply, is it kill it, fast kit and the rest, or maybe sasso and the rest? If I have one already, the first one, everyone will smell it very fast. But if I add another one, the perfume or the scent of it cannot spread very fast. Not because it's not powerful or having a high fragrance, but because the concentration gradient become a little bit lower. Because there's an existing perfume or let me say insecticide spray in the room. If I add another one, you will not smell it very fast because there's an existing concentration in the room already. So this one will make the gradient very, very slight, and that will not increase the rate of diffusion for we to smell the new insecticide spray, its fragrance. Having called Ya Anima Boatin. Anima Boatin, let me call you. I think your hand has been up for some time now. Once I called you, yes.
The state of the matter.
Okay, the state of the matter, very good. That is either it is solid, liquid, or gas. So she was talking about the condition that will increase or that will affect the rate of diffusion. That is our next subtopic. So we'll just talk about it. Now, you've all done well. All the ones that you talked to me about, they are not example of diffusion in living cells. Rather, they are life application questions, general term. Now, I want us to give examples of diffusion that occur in living cells. Yeah, Harriet, thank you very much. Your hand is up very shortly. Harriet.
Sorry.
Okay.
Gaseous exchange in the lungs of mammals.
Gaseous exchange.
So respiration.
If you say respiration, I will not take that. I will even take.
Mammals.
I will not take that. What I will take is the first one that you said. Respiration is too broad.
The gaseous gaseous exchange
Simple as that. That
in
is
the
the
lungs.
best. Very good. If we are teaching respiration, you realize that we have two phases of respiration, internal and external. The internal is what we talk about, the aerobic and anaerobic. Then under the external, that's the gaseous exchange. So if you say respiration, it could be the breaking down using oxygen. By that time, we have entered the internal or cellular respiration area. So the best one is to say gaseous exchange. Respiration is too broad. Some marking scheme will not allow you to be so generic and broad. So the gaseous exchange of carbon dioxide and oxygen in the alveoli and the lungs. Sorry, in the alveoli of the lungs and the blood, that becomes diffusion. So oxygen will leave the blood, sorry, will leave the alveoli and enter the bloodstream, and carbon dioxide will leave the blood and enter the alveoli for it to be exhaled outside. Thank you very much, Harriet Owusu Boateng. Wejem, let me call you this time. Very good. Wejem, Donko.
So please, transpiration in plants.
Transpiration in plant, very good, because transpiration is the movement of water. Did you say trans? Okay, sorry. Guttation, the movement of water. Transpiration, the movement of gaseous. It's like evaporation in those organisms. So the movement of gaseous substances out of the plant leaves and those external structures could be also termed as diffusion. Thank you very much. So evaporation or the transpiration of this substance out is also diffusion. Any other one? I think, yeah, Nima, your hand is up. You can talk to us. Wajem, you can lower your hand if you are okay, or let me do that for you. Yeah, Nima, but any example of diffusion in living cells? Once I mention your name, make sure your microphone is unmuted, because most of the times you are advised to mute it. Yeah, Nima, if you're not ready. Okay, Chantel. Okay, yeah, Nima.
The movement of gas, the gases in the stomata of plants.
Yeah, in the stomata of plant. Okay, very good. That is so the stomata, their stomata is like our alveoli. That is where they also exchange their gases, carbon dioxide and oxygen. So that is a very good example. Let me call Chantel or say to Chantel. You just joined us. Harriet has been talking. That is why I haven't called her. That's very good thing. Don't worry. I'll call you when no one else hand is up. Chantel, please, once you raise up your hand, make sure you are with the phone or your device so that you can easily unmute yourself. Chantal, I'll say you too. Okay, let me call Harry. Chantal is not ready or her network is not good. Yeah, talk.
So please, absorption of the end products of digestion
Oh, that is good, but I wanted to add.
by the small intestine
Oh,
from the
okay, very good.
That is the first.
I was waiting for this a long time. So that is what we call assimilation. So whereby the end product of digestion enters the villi of the small intestines into the bloodstream. That is called assimilation. That is also an example of diffusion in living cells. Now let's talk about plant. Okay, any other example? Yeah, Dimabwati, if you haven't talked, you can talk this time around. Harriet also Boateng, you can talk. Else I'll call Janelle. Okay, Janelle, talk to us.
Nutrient exchange.
Where? Be specific a little.
Nutrient diffusing from blood into body cells.
Okay, that's what we just talked about, the assimilation. So you just made it even simpler. Chantel, if you have another one, talk to us, then we are done. I'll give one or two, then we move on. Yeah.
Nutrients, nutrients in the cell.
Okay, well, nutrients enter the cell. That's also diffusion. Very good. So entering the bloodstream and entering the cells, these are all by simple diffusion. And there's one thing you didn't talk about. When mineral salts from the soil enters the root hairs of plants, that is also diffusion. Now, why is it diffusion? Because it's the mineral salts that are moving. But when water is being absorbed by the root hairs, that becomes osmosis. So always when water is doing the movement is osmosis. When other thing, substance is doing the movement, that can become diffusion if only we are not using energy to do the movement. Thank you very much. You've really done very well. Let's move to our screen. So now look at your screen and look at what I have projected on the screen I've shared for us all to see. Alright, so this example of diffusion, I think we have talked about all of them. So these are what you first talked about, but these are not in living cells, they are in real life examples. So in living cells is what we all talked about on the meeting as people called in to give their. So this example of extraterritorial diffusion, so these are the molecules. So you see there's no molecule here, so they can easily spread. So for instance, very good. When we are all at the A hall or in a class, what happens? When we say, okay, vacate your classes or you are from the dining hall, you are going back to your classroom, you see that since you are all together, you can easily spread around campus. But if all of you are scattered, no one can realize that students are moving in and out. So for instance, when it is break, you all come from different classrooms. Some are from the seniors block, some are from the science block, some are from the art or business block. We all meet at the tuck shop, around the dining hall and the market, different areas. So you don't actually see that students are spreading all along. But when all of you gathered at the dining hall or you are coming from the A hall, we easily spread, hey, just some 2 seconds or maybe 1 minute, all of you were at one side of the campus. In the next 2 or 3 minutes, you are evenly spread around. So anyone can notice students' movement. Like there are many and they are moving. Hey, now yes, we have a lot of students here. People can say that. But when we are all scattered from different classes, one or two people are going out for break, we don't actually feel the movement of students or the crowd or the mob of people we can have around. So that is diffusion here. You see here there are very many. So it's like you students are the A hole. Now you are moving slowly but steadily to your classrooms. A time will come, you'll be evenly spread around the campus area where we have our school blocks. This example of diffusion in liquids. So you have your maybe potassium permanganate here or you are dye in this water, you see they become evenly spread out. So this is the higher concentration, then it will now be spread around to have what we call an evenly distributed solution or let me say it, even a solution having a homogeneous mixture, let me say it that way. So here is a higher concentration, then that's a lower concentration, so they will spread around as the molecules collide, bombard, they will find their ways out of where they are crowded to where they become evenly now, these are some of the examples that we talk about. So, movement of oxygen in the alveoli, that is the gaseous exchange. This is the assimilation we talk about. That is the uh, movement of gaseous exchange in the plant cell, that is from the stomata. Now, mineral salt into water. All these are examples of now exchange of nutrients and waste in capillaries. So, someone said that is what happened before they enter the cell. They live into. Someone says she can't see. Most of us can see. So this is what you do. You can leave and join very shortly. Uh-huh. If you too you can't see my screen, give me a thumbs down. I can see from my mobile device and I'm sure others can see. If you can't see, if you cannot see, give me a thumbs down. But if you can see, give me a thumbs up. So let's do that and see. Sometimes the network can prevent you from seeing things as I do it on my laptop. Alright, so this was a hands-on activity if we are to be in the lab or in school. So you can just we have a group of members or maybe even in your house you can let your kid sister or kid brother stand somewhere, open up the perfume within some short time you all scent it. Even if your mom is cooking in the kitchen, you'll be in the hall or living room let me say, or bedroom, you will just smell it that something is cooking in the kitchen. That is the aroma from the meal, and that is through a diffusion. But now, let me ask this question: uh, when you remove a soup or any food from the freezer, and the one that you bring out from the pot, like sorry, from the stove or your microwave, which one are you likely to smell very well, and why? So a hot food and a cold one, which one do you get to smell it? Diffusion is faster. You can smell. A hand is up. Talk to me, whoever the person is. Salome, your hand is up. Talk to us. Which one and why? The one, which one?
The hot one.
The hot one. Can you explain to us why?
Sir, because the molecules are evenly dispersed and you can hear the scent well.
You can smell. The scent. Well. Okay, very good explanation, but there's a technical term. Francis Van, this is the first time I've seen your hand raised today. Talk to us, Francis Van.
Sir, please, the one from the pot.
Why?
Sir, because the molecules of the one from the pot is evenly spread rapidly because of the heat.
The heat. I wanted to bring some terms, but none of you has brought it. The collision, kinetic energy has been increased. Do you get it? So the heat will increase the kinetic energy. There is an increased collision. So once they are colliding, you see if something collides, what happens? They just spread about. So that is what I was hearing to see. That is the kinetic theory of gases. I don't know whether you've learned that in chemistry. I don't know. At first, it was in just the first 2 weeks of studies. But the new curriculum, I've not been following the chemistry now, so I don't know where it is kept. So based on that, so the heat will increase the kinetic energy of the substances. Now their collision will increase. So the bombardment will cause the rapid movement of these substances out. So that is the technical or chemical explanation to why when you increase the temperature, you increase the kinetic energy, collision happens, so they easily spread about each other. So that is why the hot substance. So it means that heat increases the rate of diffusion. Thank you very much, Francis, Bai, and all the other people that have talked today. You've really done well, very, very well, and let's keep it up. So that is, you can put your perfume over there, you smell, or you can put a potassium permanganate or any colored substance, put it in water, just watch it spread. Now, what about what we call blue in Ghana here? That is our potassium permanganate whatsoever, the blue band and some other brand. We call some Abidjan blue. I'm sure all of you have heard it before. In the SS our time, if you don't take care, you now have some specks, or let me say some dots in your eye shade because you didn't apply that Abidjan blue very well. So the granulated, when I say granulated, the one that are having small in particles. So the granulated potassium permanganate and the powdery one, the one in that blue box. Which one do you think, when you put it in water, can evenly spread or diffuse in your water, and why? We are now tackling factors that affect the rate of diffusion. We have now realized that heat is one. Someone just raised their hand. Who is that? Harriet also, today you are doing very well. Please, the rest, I encourage you to also be talking. It seems today I'm having about 5 students doing the talking. Okay, Harriet, since your hand is up first, I have nothing but to call you. Yeah.
Please, the powdered one.
And why? You add why?
Um, the powdered one has a smaller molecule, actually.
Smaller particle
The
size.
size of
Yeah.
yes, the size of the diffusing particle is also a factor. So when we have a smaller molecule, it moves faster than
Very good. That is so perfect. So, Chloe, I'm sure you would have said the same thing. So, next time I'll call you because of time. So, the particle size, as you said, now you cannot see my screen. You can now see my screen. So that the first one is concentration gradient. So the higher the concentration gradient, the higher the rate of diffusion. The lower the concentration gradient, the lower the rate of diffusion. So take that one very well. So if they ask you mention things that affect rate of diffusion, after mentioning the factor, try to tell us why that factor or how that factor increases the rate of diffusion. As if your hand is up again, what do you want to tell us? Do you want to explain something? Okay, thank you very much. You realized the hand was up unknowingly. So the first one is concentration gradient. The steeper or higher, the higher the rate of diffusion. Second is temperature. We have explained that already. Francis Bar and the other ladies also talk about it. So the higher the temperature, the faster the rate of diffusion because heat will increase the kinetic energy, which increases the collision of the particles or molecules. Thereby, they spread out very fast. So that is it. Now, temperature. Sorry, we talk about that. Now, the particle size. The larger the particle size, the lower the rate of diffusion. So it's like we call this thing adanchene, that granulated salt, that particle salt. Instead of having the anapuna and the other iodinated salt, you see when you put that one in your soup that is not on fire, you don't take care, you'll be taking particles from under your pot. But when it's on fire, you don't even stir it, it will just diffuse off. So it's like putting salt or sugar in a hot porridge. You don't have to be stirring it or shake it, it will just diffuse. But if you put it in a cold Tom Brown, you will see what happens. If you don't stir it, you will eating sugar at one end, and the other side you'll be eating the raw Tom Brown or porridge. So these are the factors that affect the rate of diffusion. Chantel, I'll let you come in for now. I don't think I've asked the question. So Chantel, you can keep your hand up for the next question. The next one is the surface membrane. If you have a question too, I'll let you ask your question. You can keep your hands up. Now we call something surface membrane. So the particle size affect the rate of diffusion. Now surface membrane. When we say the surface membrane, it's talking about, for instance, it's like having a paper and one that is not folded. Let me use this for instance. So now this is like the entire surface membrane of this paper. If I try to fold it this way, which way can water diffuse into this very faster? This one, it can't diffuse well. So that very good. Having a camphor. When you buy a camphor, now we have very, very big ones. If you grind it, you have increased the surface membrane. So it will spread very fast. You will smell the scent very, very fast. And within a day or two, all of them will vanish. But if you put that particle form over there around the corner of your room or in your bag, it will take several weeks or months before it all diffuses out because the surface membrane is still small like this. But if you crush it into granules, or let me say into smaller granules or into powdery form, it becomes like this. Now what happens? It can easily diffuse. Let me talk about the way we hang our clothes. When you want to wash your clothes, sometimes you spread it out in the basin or your washing bowl so that water can absorb very well. But if you squeeze it something like this and you put it inside your washing bowl, it take a very long time for water to absorb to absorb water. So and if you're hanging it, what do? You spread it up, you put your pegs at some ends so that water will drain, and as the wind blows it can easily dry. That is also diffusion. So okay, let me say it won't be diffusion, but I'm just as an example because water is rather leaving it. So once you hang it, it has a higher surface membrane, so it can easily get dried as the wind blows. So that's the surface membrane. So the thicker the surface membrane, the slower the rate of diffusion. So if you have the membrane like this, very, very thick, it can't evenly distribute because the particles will not be allowed to move very fast. But when you open it up, a larger surface membrane, it easily dries up or diffusion occurs very, very well. That is why if you are using the sugar cube, now we have that sugar cube. When you put it in your tea or any other food substances, you need to stir it. I know some students, if they want to do it very fast, they sometimes we even have to crush it. You put it in your cup or your container, then you just crush it. You just break it down before you add your Milo or whatsoever. But if you are using the normal granulated sugar, you just put it in and you are on the go. So that is the surface membrane. You see, when you take the sugar cube, it's something like this, very, very smaller. But when you crush it down, it spreads out, then easily it can dissolve in your food. Let me take the last two or three. You have a question you can ask. Now, stirring. We talk about that. If you want that your sugar to spread very fast, you stir. You stir it very fast. So why are you stirring? Stirring also causes an increase in their movement, as heat does. Heating will increase it by increasing the kinetic energy or their heat energy, so they will collide very fast. That is what heat does. And if you are also stirring manually, you are increasing their collision, so you make them collide. And stirring also break them down. As you stir something, you break it down. So if you put your meat in your stew and you stir very vigorously, you will end up seeing meat mache. So your meat will rather become very very smooth. But if you don't stir it that wide, you still have your meat. So sometimes your parents tell that, oh, don't stir the soup like that, because you'll be breaking salmon and some other fragile meats in the soup. So stirring also break them down. It also try to increase the movement or collision, so they can easily spread out in your substance or the space. Now, the next one is distance traveled by molecules. I'm sure when you are in classroom, hardly do you smell from the kitchen, especially those at the visual art block. If they're cooking something from the dining hall at the top over there, that is where they have our dormitories. Hardly will you smell. Now that you are in your house, you can't smell something moving on in the next three houses unless they are cooking it outside or something like that. So the distance, if I apply my perfume here, no one else on the call can smell it. You rather see me applying my perfume or fragrance, but you can't smell because of the distance. So the distance you will travel also affect it. So sometimes you go to the refuse dump, or if the public toilet, if you are around that area, you smell the foul scent. But when you move away from that area or from the toilet, even on campus, you don't smell it well, or you don't even smell it at all, because the distance, the that scent is not much to spread. But when it becomes so much, it can even spread to a longer distance. So the higher the concentration, the higher the distance it can cover. And the longer the distance to cover, the slower the rate of diffusion. So under the distance traveled, the shorter the distance, very fast. So if someone apply a perfume in your class, it spread very fast. But if the person is at the A hall, or maybe let me say Kumasi Sports Stadium, someone apply it, even a market, you see that the distance is very wide, so it cannot spread very fast as you are in a dormitory or in a cubicle of any of the prefects on campus. So these are the factors that affect the rate of diffusion. At this point in time, I will give you opportunity to ask questions if there is any. If you have a question, we have 20 minutes to end the lesson. By 9 o'clock, as I've been telling you, I would like us to end the lesson so we don't take my time of your night. I'm sure it's very clear, everyone understand. I'm waiting for the next 5 seconds. If no hand pops up, I will be teaching the next section. Okay, thank you very much. There's no question. If there was, keep it for the next question and answers section. Now, this is an example of diffusion as I talked to you in living cells. We've talked about this thing, so no, we didn't talk about movement of hormones out of the endocrine glands into the blood. It's one example we didn't talk about. You can add that one to your examples of diffusion. And also unicellular organisms like the amoeba, parasmecium, euglena, and spirogyra, they also take in water by simple diffusion. Sorry, they take in carbon dioxide in, out, and oxygen in by simple diffusion. So these are some examples of simple diffusion. And I think we've talked about this already, so let me move on. Now the next one is osmosis. We have defined osmosis. So I've told you that osmosis is the movement of water molecules. So when the water is moving from a weak solution, if you don't want trouble, just say weak solution. Don't say from a region of higher solvent concentration. You might end up confusing solvent with solute, and that will make it wrong. So once we say from a weak solution, that is all. A weak solution is a solution whose water content is larger or higher than the solute concentration or solute amount. That is osmosis. Now, based on osmosis, we can have three terms. Let me check whether we have it here. That is osmosis in cells, very good, is here. I'll talk about it very shortly. So let me take it gradually. Now, the factors that affect diffusion, I would say all of them or most of them affect the rate of osmosis. So here, let's look at something here. We call something factors that affect the rate. Then I'll talk about the hypotonic and isotonic and the rest. So we see when you increase the temperature, it also increases the rate of osmosis. When you talk about concentration gradient, the higher the concentration gradient, the higher the rate of diffusion. Sorry, osmosis. Sorry. Right now, if I say diffusion, I mean osmosis. So remember, we did this example in junior high school. I'm sure most of your teachers guided you to do it. You make a hole in a yam cavity or a pawpaw cavity. You put it in a basin containing water, and you put some salt inside the yam or pawpaw cavity. What happens? The next day, overnight, you see that water has moved from the basin through the pawpaw or yam cavity, that is the semi-permeable membrane, onto the space you made or the cavity area. Why did it happen? So the more the water that moves, it means you had enough salt or sugar inside the cavity. If you put in just one tea, what do you call it, 1 teaspoon over there, you will not have enough water. If you put a cup of water in the cavity, you realize that when you don't take it, the next day all the water in the basin will move into the cavity. So the higher the concentration gradient, the higher the rate of osmosis. And the permeability, remember, under osmosis we are moving through a semi-permeable membrane. So if that membrane is not permeable, it can't move. That is why you didn't use a dead or a cooked or boiled yam. You are asked to use a fresh one because the cells are still living in. They are not now starchy or dead, so that they can allow for the movement of the substances. So we have to talk about the permeability of the food, sorry, of the substances or the area or the region that the osmosis will occur. Now, application of osmosis in everyday life. Let's look at example of osmosis. So let me close what I have here. You can open your microphone. Tell us some example of osmosis, either in real life or in living cells. Let's put them together. I want in living cells or in real life. Example of osmosis. You can raise up your hand at this time. We have 15 minutes left to end the lesson. So, example of osmosis. Yeah, Vanessa, chance for thanks for coming in. Talk to us, Vanessa. Yellow. Yeah, talk. I think I'm getting. I don't know whether where you are is very windy. I feel. I don't know whether it's a fan or something. I'm getting some echo and feed. I'm sorry. So you are giving me a lot of background noise. So Vanessa, you type it. Sorry. Let me mute yourself. I don't know. The noise is too much. I'm getting a lot of noise in my. Thank you very much. If you can't work on that, you can type it in the comment section, in the messages. Okay, so yeah, Nima, let me call you. Ask Vanessa work on her microphone and her background noise for me. Yeah, Nima, what's in?
Plant absorbing water from the soil.
Yeah, plant absorbing water. Or so to make it very simple and specific, whereby the root hairs of plant absorb water. That is very perfect example. Salome.
Say dry fruit rehydrating in water.
Very good. So when you put your date seed or maybe sometimes cassava in water, you see that water will move into the cassava. You ask yourself, what moved? Is the water? That is osmosis. Very perfect. You've done well. Uh, I don't know. Did I call Salome or Agnestevia? I'm calling the other one.
Salome.
Salome, I've called you.
Yes, sir.
Okay, then Agnestevia. So please let me lower your hand for you so I don't call you again. Agnestevia, if you are ready, talk to us. Example of osmosis in living cells. Okay, Chantel, your hand was up long time. Let me call Chantel now. Chantel, say it to you. So I didn't hear you. Come again.
cucumber in brine.
Cucumber in brine. Okay. So here, where does the water move to? Because it said brine. Where is the water moving to? Did you say yeah?
Yeah,
Okay.
no, it's not true.
So what's the answer? Can you help us?
And the water moves out to balance salt concentration.
Very good. That is perfect. So in this case, water will not enter the cucumber because it is brine. So for those of us who are confused about brine, brine is a salt solution. So depending on the concentration of the salt in the water, water will move from the cucumber into the solution because water will move from where it is more to where it is less. So since it is brine, water is lower. The concentration in the salt solution is higher than the cucumber. So water will leave the cucumber into the solution outside of the cucumber. Thank you very much for that. Hey, Mobina Mokhtar. Sorry if I couldn't pronounce your name well. Mobina.
Sir,
Yeah.
please, salty food draws water out.
Yes, salting food. Okay, so water will move out of the salted fish, or you say Kobe. So that is osmosis. But this any other hand I can't see it now. I'm waiting for it to show on my screen. A new hand up now. Esther, someone here, and some other people. Esther,
Movement of water into cytopla- cytoplasm of amoeba.
of amoeba, very good. We will learn about them. So water move into the body of amoeba. Since water is moving, we will not call it diffusion. Amoeba too can engulf food, and that becomes by phagocytosis. So amoeba feed by phagocytosis. But when mineral salt and some other disorders are entering it, it could be diffusion. So carbon dioxide will enter, will leave through diffusion. But food, because it will engulf the food, it will rather be phagocytosis. But oxygen will enter through a simple diffusion. Janelle, you are in again. Janelle, talk to us. Just one or two, then we continue.
Movement of water from root hairs into the cortex of
Yeah,
plants.
thank you very much. Salome, if you still want to talk, the last one, then I will lower all of you your hands because all of you have talked. So Salome,
Sir,
yes,
sir, please. Our skin wrinkling in a bath.
our skin
Yes,
wrinkling
in a bath.
in the back.
In a bath. BATH.
BAT. Okay, 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 okay. Okay, so it depends on the type of bath you are having. If you are having, I don't know, substances in the water like salt and brine, then water can leave your body cells into the bath. So I don't know the kind of bath you are having, but if it's just normal water, your body will not actually lose a lot of water because the concentration in your body might be higher than normal water. So it depends on the substance you have in the bath. But if it is salty water or brine, then water will leave your skin cells into the water bath you have in your jacuzzi or whatsoever. Thank you very much. Thank you very much to all of you. If there's a question someone can ask, if you have a question, Salome, you are very good. If it's a question, you can raise up your hand. But if you want to answer my questions, thank you very much. I think we are done for that. If you have a question, you can ask. If not, let's the next 8 minutes to talk about something else. Okay, no hand is up. Now let's move to our slide. Now watch my screen. Watch my screen. Let's talk about something very shortly. Now, let me finish this. We call something osmosis in cells. Due to difference in concentration and the movement of water in and out of a cell, three things can happen. Okay, let me say two things can happen. And these two things that do happen, they can be broken into four. Now, animal cell, we know animal cell don't have a cell wall, but plant cells do have a cell wall. Due to that, when water move into an animal cell, in excess, something can happen to it, which is different from what happens to the plant cell when it is in excess. So we have three main types of solution. Now, either you are equal to me, I am above you, or I am below you. These are solutions. So we have isotonic solutions from the word iso. And since we don't have time, I will just say things myself because of time. Iso from the word isotope. Normally, I will let my students define isotope. And remember, they will talk about someone want to define because of time. I will not allow you. So they are atoms of the same element with the different mass number, but since they have the same atomic number but different mass number, due to the differences in their neutron number. So the reason is we are saying the same element, same. So iso means same solution concentration. Then we have hypo. Hypo means low. The other time I talk about hypoglycemia, whereby your sugar level becomes lower than the recommended 5.8 millimole around that area. So when you drop somewhere 2.3, 4.5, you are having hypoglycemia. Then we call something hyper. I'm going to a supermarket, a hypermarket, a hyperbole. So hyper from the word hype. If you're hyping something, you are making it bigger. You are making it bigger to trend. So hypertonic solution is a solution whose concentration is greater than the one is being compared to. So for instance, if we are preparing a tea, I'm sure most of the times your kid sister or brother will make his very very sugary, and your father or mom will not even add water to his or hers. The issue is when we are comparing the tea that your brother or yourself you made compared to that your grandmother or your parent, you see that yours will be too sugary than that of your parent. So your tea or your solution is hypertonic to that of your father, because maybe at that age, normally when they are getting old, we become too careful about health. Either the person is diabetic or they are trying to prevent those metabolic syndromes. So adults don't take enough sugar unless the the recalcitrant one. So most adults, their sugar level they reduce. But kids, they want to taste it. Their taste buds they are very active. They want to enjoy their taste. So that of your tea compared to that of your parent, yours is hypertonic to that of your parent. Now, in the other or the vice versa, that of your father or mother is also hypotonic to yours. So if I am hypertonic to you, it means you are hypotonic to me. That is a reverse. Now, if what you prepared, you give same one to your junior brother or sister, it's the same concentration, the same taste. So yours and that of your sister or sibling, other sibling, is isotonic. It means it's the same concentration. So you added 3 teaspoons, you also added three, and the same water, the same milo amount. So you and that your brother, you have the same concentration. So that's isotonic. Then we have hypertonic and isotonic. If you understand this, give me a thumbs up, then I'll move on. Now, animal cells have only cell membrane. They don't have a cell wall, and plant cells have a cell wall. So due to that, something different will happen to them. Very good. Edna says she understand. If you don't understand, give me a lower time. If I see only one, I'll even explain again. So we have isotonic, hypotonic, and hypertonic. Very good. Now let me come here. Someone says she doesn't understand. Esther Kumache, you just joined. I saw you living and joining very, very of late. So I'm taking just 2 seconds. Try to understand, or you rewatch the video when I upload it on the channel. So we said isotonic solution. Iso means the same. Someone says she doesn't get it. So sorry, today we might go some few minutes beyond the 9. I have to finish this. So please, some are saying they don't get it. So I will take my time and explain again. So if you can hear me, I said based on the concentration of solution, we can have three comparison. Either you are having the same concentration. So for instance, if you prepare a cup of tea and you stir it very, very well, and you give the same to your brother or your sister or your friend, it must taste the same, because you prepared it in the same bowl, you stirred the thing, the same amount of sugar, the same tea that you added, let me say coffee or whatsoever, then you shared it with your brother or sister. The concentration or the taste, the sugar level will be the same as that of your friend. That is what we call isotonic solution. If you are soup, you prepare a soup and you added enough salt, you stir it well and you share it in the family, your father shouldn't complain of salt. Yeah, yes, he will complain of salt because he is old, he doesn't take enough salt. But when your brother want to confirm, you say, ah, daddy, but yours is the same as mommy or akosia, because it's the same food. But because for them they don't want to take enough salt, they will say it is salty. But when everyone tastes, they will say that, ah, it's the same as that of yours. That is isotonic solution. So solution whose concentration are the same is isotonic. If you understand now, let me know. Just thumbs up. Don't bring your microphone. Just thumbs up. Then the next one is hyper. Hyper from the word I use hype. If you're hyping something, you are making it very elegant, very larger. People, you are making it trend. So hype means you are making it greater. Announce it. So hypertonic solution is a solution whose concentration is greater than the one that it is being compared with. So if I am comparing that and I use a simple example, you are making a tea. Most often than not, your porridge will be sugary than your parent because you are young, you want to enjoy, have the taste. But your parents, they are older. Our parents are old, they don't take in a lot of these things. Sugar or salt, if you put in small salt, they will say that, hey, it is salty. They wouldn't take it because they are very careful about their health. They don't want to get hypertension and diabetes, metabolic syndrome underlying. They don't want to get them. But you, now that we are young, we want to enjoy the soup with enough salt, or maybe the porridge with enough sugar. So if yours is sugary than that of your another person, yours become hypertonic because your concentration is greater than the other person. That is hyper. So if you are greater than me, then I am lesser than you. So that of your parent whose sugar content is very low, his food or his porridge is hypotonic to yours. Esther Kumache and the other person. Now do you understand? So I can move on. Esther Kumache, you can give me a thumbs up or you can open your microphone and talk to me. Someone is making noise. I want to mute that person. Esther, do you understand now? Okay, I'm sure by now everyone do understand. Okay, let me continue. So we have isotonic, hypotonic. Chantel, if you can't hear me, what others can do, you can leave and join again. Princess, you are talking. What is it? Okay. Our time is up and I don't know whether we can have some. If not, I'll let us end it tomorrow. We continue. I don't think still we can even finish it. So I would like to end it here. If you have a question, ask. Since tomorrow we have a meeting, I will leave it there. We continue from there. If there's a question, ask. So please kindly read this. You talk about hemolysis, turgidity, and the rest. So now you talk about diffusion osmosis. This is the last bit. Osmosis in cells. Our time is up, so we will end the lesson very shortly. If you have a question, you can ask. So tomorrow, God, that's our lesson to talk. Any question you can ask now. Okay, since there's no question and our time is up, I don't want to take much of your time. I'll let us obey our time. It's 9:02. So God willing, tomorrow we will talk about this isotonic, hypotonic, and okay, someone want to view. They have a question there. You have it already. I've been sharing this document with you, girls. So it's on the WhatsApp platform in the Google Classroom. You have it. So the one that okay, who is the one talking? Who says she doesn't have it? Let me show you where you can get it. Who is the one talking with me? Salome, are you the one? Okay, so If anyone doesn't have any material, let us know. It's on the Google Classroom platform. Periodically, I share it on the WhatsApp platform for late members. So very good. Our next subtopic is we will talk about the osmosis in living cells. So based on that, when enough water entered animal cell, it become what we call ash. It will swell. When it swell up, later it become hemolyzed or it will burst. I'll explain this to you tomorrow because of time. If I'm to do this, we can go extra 30 minutes, which I don't want us to go deep into the night. So I will talk about this. So table 3.1, conditions that result as water move in and out of cells. So we look at what happens to animal cells and what happens to plant cells. So water moves into the cell, water moves out. Some will become flaccid, some become plasmolized, and the rest. Don't worry, I will explain that tomorrow. Then we also talk about this hemolysis. I will explain those terms. So please read it. There are some keywords you might not understand. I will explain tomorrow to you. We'll talk about flaccidity, crenation, plasmolysis. Then we will start active transport. If we have enough time, we will talk about the vesicular transport, and I will explain this to you when I was talking about the endocytosis and the endocytosis. So this is what we will continue tomorrow. I am praying we have enough time to finish week 3 lesson so that next week Sunday we can start our week 4 lesson. That is sorry, section 4. That is week 6 lesson, ecology. Then bit by bit we can finish our syllabus. Now let me remind you, once we resume on March 16th, we are writing our end of semester. So please take this very serious. You know that for some subjects and for some schools, I've been contacting some other schools, they just submit the documents there and they are to read. Yeah, I understand. You should be able to read on your own. But I think this will be an extra for you people who are joining. So make sure you take everything serious. If you want to rewatch this, I will later edit and upload on the YouTube channel and you can benefit out of it. So that is the announcement for today. Covering tomorrow same time, we will join and have our continuation of the lesson. So keep on learning, not just biology, but learn the other topics as well. If there's any other issue, you can reach out to the administrators or personally to me. When I have time, I will address your issue. So have a very good night. Thanks for being my student on this virtual platform for tonight. So we will meet on the other side. Bye to you all. You can start leaving the meeting or I'll end the call so that we all leave. Have a good night. Bye to you all.